Hey everyone, this is our lesson on microscopes, and by the end of this lesson you should be able to use microscopes to observe a variety of cells with particular emphasis on the differences between prokaryotic and eukaryotic as well as plant and animal cells. So we should be able to see the specific structures that classify or identify those types of cells. So first let's get into microscopes themselves. So these are machines used to form an enlarged image of an object or specimen because there are certain things that we just cannot see with the human eye. So we have to have some sort of magnification to make that possible. These are also used to observe living organisms or preserved cells. So when we do a lab, we're going to look at mostly preserved cells, but we could look at some living organisms as well. There are two basic properties of microscopes to know, and that is magnification, which is how large an image is that you produce with the microscope, or resolution, which is how clear the image is. So for magnification, we look at low and high powered most of the time. So low powered magnification is about 100 times the size of the specimen that you're looking at. And then high-powered magnification is about 400 times. And then, like we said, resolution is how clear an image is. And you have better resolution on a high-powered field, but the view, like the field of view, what you can see is smaller. So if you look at this GIF over here, we show a full cell, but as we zoom in with a stronger lens, we can see more clearly smaller objects, but now our field of view is limited to just the DNA in the nucleus as opposed to the whole cell, which is what you see from far out. So just as a definition, field of view is the area seen through the microscope. So it's what you can see before the blacked out edges. We cannot see as much of the image on a higher power, but we can see more detail within the portion that we can view. So the type of microscope that we come in contact with most often that you be using in this class is a light microscope. So this uses the lenses to magnify the organisms or the objects you're looking at. So a compound microscope has more than one lens, and they can magnify objects up to around 1,500 times the original size. But the ones that we tend to use in schools are around 400 times max viewing. So we have three different magnifying lenses that we will look through. I think it's 10, 100, and 400 that we kind of can view these at. So higher magnification is more expensive, which is why us in a public school do not have higher magnification. And light will pass through two lenses. So first it'll pass through the objective lens, which is the lens closest to the object. And then we have the ocular lens, which is known as the eyepiece. So that's where you look through. Now, we don't only just have light microscopes, but in more well-funded labs, higher education labs, you have also what are called electron microscopes. And these are really cool. So light microscopes are really nice for seeing small organisms, but these give the coolest looking views of organisms or whatever you happen to be looking at. Just like this GIF here shows like a picture, or not a picture, but a flea, and an electron microscope zooms all the way in to something that's located on the eye of a flea. So that's how far the magnification can go on some of these. So we have two different types we're going to talk about here, just so you know how advanced microscopes have gotten over time. And the first one is a transmission electron microscope. And this aims a beam of electrons through a specimen, and it'll magnify objects hundreds of thousands of times so we can see the tiniest of objects. Then we have scanning electron microscopes, and these use electrons to scan the surface of a specimen, and they produce a 3D image, which is more what we're seeing over here. So you can see at the bottom that it says SEM, that is scanning electron microscope, because it uses electrons to hit the surface and to give us a detailed outline of what we are viewing in a 3D image. And this can actually magnify up to 60,000 times the original organism size. So when you're using a microscope, there are certain steps to go through. So first off, I do want to address this GIF over here. When you are carrying a microscope from its home to the lab space that you need to use it, there is a specific procedure to do this. There is an arm and a base on the microscope, which are pointed to in this GIF. You always grab a microscope with two hands. So you hold it by the arm and the base and you carry it to and from, just so you know there's no way to damage the eyepiece or the lenses. So after you've done that, you will place the slide on the stage by taking the slide off of this and taking the slide off the stage. You need to make sure you return the lens to the scanning objective. So you want to get it to the lowest viewing and you want to get it lower on the stage. So you want to use the knob to lower the stage down. You don't want to scrape the slide with the lens. After locating the specimen, it must be centered before you change to a higher powered lens. So you do want to move the slide as best as you can to get the object in question in the center. And then before focusing on the high power on the specimen, you must focus with the low power. So you start with the lowest, focus, move it up, focus, move it up, focus. Never use the coarse knob with the high power lens. So the coarse focus knob is the one that will move your base up and down. Then you have a fine focus knob. I think there's different names for that. I've always just called it a fine focus knob. A fine focus knob that will move it very, very small distances that you use in terms of focusing with that high powered lens. So you'll never turn the fine adjustment knob, there you go, more than one full turn forward or backward. So if you're having to move it more than that, then that means the coarse knob should be used before that. And then to put this away, you put the scanning objective lens in place, so the lowest one. You'll turn off the power, then you unplug it, you clean off the stage and the slides, you place the microscope cover